Hi everyone, we are back with a new topic related to your geology syllabus. Today we are going to discuss the basic principles of protein sorting, or also called protein targeting. Let me first tell you what is the present idea of how the organelles evolved in our cell. In this slide, you can easily see that there are two different kinds of cells at these two extremes. The one is called prokaryotic cell and another is called eukaryotic cell. So in between there might have been several intermediates, which are of course possible intermediates. So how the eukaryotic cells, which contain membrane bound organelles, that evolved from a simple prokaryotic cell. It is assumed that the possible intermediates from where the eukaryotic cells are presumed to have evolved might have certain regions on their plasma membrane that used to serve for protein secretion or protein targeting. Now these specialized regions might have been invaginated in the course of evolution, and they became more specialized and they gave rise of the intracellular organelle. For example, nucleus, endoplasmic reticulum, Golgi apparatus, etc. Now in the case of eukaryotes, what happened that rather than being secreted, these proteins after they are synthesized in the cytoplasm, they now used the channels, for example the Golgi apparatus, etc., through which the proteins are sorted and targeted to the plasma membrane or even outside the cell. Now there are thousands of different kinds of proteins which are synthesized inside the cell, and they of course have different functions based on their location. The function of a protein is location dependent. For example, a protein which is supposed to be in the nucleus must be in the nucleus in order to function properly. And then there are proteins which reside in the Golgi apparatus, there are proteins which reside in the rough endoplasmic reticulum. So the proteins needs to reach their proper destination. So the proteins, once they are synthesized into the cytoplasm, called to the process of translation, then they are delivered to their proper destination. They might reach the lysosome, they might go to the mitochondria, etc. So this delivery of newly synthesized protein to their proper cellular destination usually referred to as protein targeting or protein secretion, protein sorting. Now the major events which are involved in this protein targeting is the targeting itself. So here in the first part of targeting, a protein, when they are synthesized using ribosome, they are targeted to a particular protein channel called translocone, through which this protein gets the access of the lumen. So this part is called the targeting, and then there comes the second part, which is called translocation. So here the protein is physically moved from the ribosome towards the lumen of the endoplasmic reticulum, and now inside the endoplasmic reticulum these proteins are covalently modified, so they are glycosylated and other modifications are done, which we are not going to discuss in this topic. Now for the ease of understanding, we have categorized these secretory pathways into two different groups. The first group is called non-secretory pathway and the second group is called secretory pathway. The non-secretory pathway of protein sorting is that pathway where the proteins are synthesized in the cytosolic ribosome and they either remain in the cytoplasm or they might reach to a particular organelle. For example, it may be a mitochondria, it may be chloroplast in case of plant, it may be peroxisome, or even it may be a nucleus. So this particular pathway is called the non-secretory pathway. Now in the secretory pathway of protein sorting, the proteins are synthesized on ribosome which are present or which are studded on the endoplasmic reticulum membrane, outer membrane. So while they are being synthesized, they are imported into the endoplasmic reticulum and followed by they are translocated to the Golgi complex. From there they can either reach the plasma membrane or they can even reach the lysosome. So this particular segment or the part of the protein secretion is called secretory pathway. So for secretory pathway they use a different sequence called signal sequence which make these proteins go into the organelle like Golgi or even lysosome. In order to now understand the principles of protein sorting, we will take the example of a protein or the polypeptide which is destined to reach to the lumen of the endoplasmic reticulum. Now what happens is that during the translation a sequence is exposed, we call it signal sequence, because this sequence can bind with a protein called signal recognition particle. So this particular protein can recognize this part of the polypeptide as a signal, and then upon binding to this signal recognition particle or the SRP, this SRP along with its polypeptide, it gets bound with the SRP receptor which is present on the lipid bilayer of the endoplasmic reticulum. Now this brings this polypeptide into close proximity with this translocon channel that is present on the vicinity of the SRP receptor, all of which are present on the lipid bilayer of ER. Then this polypeptide gets the chance to move through this translocon channel with the help of the GTP expenditure. So the question is, what is a signal sequence? A signal sequence consists of 16 to 13 amino acid residues present either at the C-terminal end or at the N-terminal end. However, the signal sequence of the endoplasmic reticulum, those polypeptides contain this signal sequence at their N-terminal end. Now there is no apparent similarity among the different signal sequences present in different polypeptides. What similarity is present is that they have a hydrophobic core followed by a positively charged amino acid either at the C-terminal end or at the N-terminal end, or even at the both C as well as N-terminal end. Interestingly, in most of the cases, these polypeptides doesn't require this signal peptide for their proper functioning, which in other words means that these signal peptides are removed upon reaching inside the lumen of the ER. So in the functional mature protein, you will never find these signal peptides present in the protein. Gunther Blobel is the scientist who discovered this mechanism of protein sorting and gave the signal hypothesis, and his discovery was recognized in 1999 as he received the Nobel Prize in Medicine or Physiology for his discovery. In order to understand in greater details how these signal sequences help this protein or the polypeptide reaching the ER lumen, we'll have to see this diagram here. This is a co-translational process, which means that while being synthesized this protein, the signal sequence which emerges from the nascent polypeptide is captured by a protein called SRP, as we indicated in one of our previous slides. Now this SRP, what happens is that it gets in touch with the SRP receptor which is also present on the lipid bilayer of the ER, and now this binding is strengthened with the binding of the GTP with both the SRP as well as the SRP receptor, and this GTP are hydrolyzed and the energy is utilized to push this polypeptide into the ER lumen. Now as you can see, upon reaching to the ER lumen, so this particular signal peptide is cleaved with the help of an enzyme called signal peptidase, which is present on the inside of the bilayer of ER. Now this functional this protein, this polypeptide, no longer it contains the signal peptide because it is already clipped, and now it gets the opportunity to fold properly so that it can function. This translocon channel consists of a total of 3 different proteins. The one is called Sec61 alpha, and there are 2 other proteins called Sec61 beta and Sec61 gamma. So together they make this translocon channel. Now in order to properly function, so this translocon channel must bound with the ribosome so that the polypeptide can enter into the lumen of the ER. So if there is no ribosome, the channel remain closed. That's all about the secretory pathway of protein sorting. If you think this video is useful, please like this video, subscribe my channel and hit the bell icon to get newly posted video.